So we'll just carry on then. So we left off talking about acids and bases, right? And so we talked about the dissociation constant for hydrogen and hydroxyls. You guys remember what that is? And there's so remember we have water, and water, the oxygen doesn't share very well, so the electrons spend a lot of time, and then this will dissociate and make H plus, and then this makes OH minus, and that no matter what solution it is, these multiplied together have to be 1.0 times 10 to the minus 14 molar. Alright, so in pure water, if this is made, that has to be made. One of those is made, one of those is made. That means that these have to equal each other. So what's the value of these in pure water then? So this one's 1.0 times 10 to the minus 7th, and this one is also 1.0 times 10 to the minus 7th. I'm just going to stop writing molar. You guys understand that that's what it is? So when we multiply exponents, you add them. So negative 7 plus a negative 7 is negative 14. Okay? So so pH is power of hydrogen. So we're interested in this. That doesn't matter. But we can use this to calculate this. So what's the pH of pure water then? It's this value. Drop that. So it's 7. So what's the pH of this then? If I tell you that that is the concentration of hydrogen ions in a solution, let's say a lake or a river or your drinking water, so this would be 1.0 times 10 to the negative 5th, right? Because we moved the decimal, remember we learned this the very first lab. So then this, because that's H pluses, what's the pH? No. It's this value, right? So would that be an acidic solution or a basic? It's acidic, right? Because the rule is if the H plus is higher than the OH, right? Then it's an acid, and if it's lower, it's a base, right? And the pH scale is a little weird because which one of those is bigger? This one's bigger, right? But if we convert that to whole numbers, which one of those is bigger? Yeah, so it gets a little confusing for students. You just have to remember that the pH value is based on this value. And as this goes smaller, this is actually, right, so it's increasing as we get smaller exponents, it's increasing the number of H pluses. So it's becoming more acidic. Yeah, if the negative value is, but the whole number value is greater than 7, then it's a base. Yeah, that's right. Alright, so On the test, I might say you don't have a way to test the hydrogens, but you determine that the OH concentration in a river is that. What's the pH? So that's 1.0 times 10 to the negative 6. So the pH is no, no. Remember, pH is power of hydrogen, not the power of hydroxide. So if that one's negative 6, then what's the H? Because they have to multiply together, they have to equal 10 to the negative 14. So that means that the pH is 8. That's the correct answer. That makes sense? All right. So an acid is defined as a substance that increases the number of H pluses in a solution, because that's the only way to make something more acidic, right? If you want to make your stomach more acidic, you can add H pluses into it, and that's what your body does. It takes them out of your bloodstream and sticks them inside your stomach. If you want to make it less acidic, what do you have to do? Well, I mean, ultimately, what do you have to do? No, simpler than that. Take away the hydrogens, right? So pH changes by the increase or decrease of hydrogens. That's all it is. There's nothing else you can do except increase or decrease hydrogens to change the pH. Okay, so let's say that I get a bottle of muriatic acid, which is hydrochloric acid from the pool store, right? And I pour that in my pool. What am I doing? I'm adding H pluses. Well, how does that work? So what's hydrochloric acid? It's H plus, and then Cl is minus. Which one is the cation? Which one's the anion? Right. So yes, that's right. This is the cation. That's the anion. Good. So when I add this in to water, I'm directly increasing the number of H pluses. So what happens to the pH number? It goes down because it's increasing the negative exponent, right? Remember we agreed that that is bigger than that when we're talking about to the raised to the 10 power raised to that exponent. Make sense? All right. So no matter what substance it is, if it is increasing the H pluses, we call that a donor because it's donating protons to the solution. So an acid is always a hydrogen donor. Bases work by doing the exact opposite. So what do they do? They decrease the H pluses. So you guys probably don't remember this, but this is ammonia in H3, right? Remember when we played that with the chemistry set and the the amino, the blue ball had like 5 holes in it, but I told you normally it would only have 3 reactions. So, and it's because it can lose 3 electrons, gain 3 electrons or lose 5 electrons. Sometimes it gets confused. But anyway, so in this case, ammonia can actually add a hydrogen to it, and then that becomes an H4 plus. So what has it just done? Right, but in the solution, if I opened a bottle of ammonia and I poured it in my pool, what would it do? take away hydrogens, right? H pluses. So what is that defined as then? It's a base. So a base is always an acceptor of protons. That's the term we use. Does that make sense to everyone? All right. So, well, yeah, but where's the H plus coming from? It's coming from our solution, right? So if I have water, let's say I have pure water, and I'm pouring my ammonia in there, remember water dissociates at a certain rate, that's coming out, that's where that H plus is coming from, right? So it's taking H pluses out of water, which makes it more basic. Right. So the pH go up or down. The number goes up, right? The negative value, 10 to the negative value goes down. That's right. So this is directly taking H pluses out of a solution. But there's another way you can do it. So we have sodium hydroxide. You guys know the charge on sodium? And OH. Remember we have H2O. So the electron stays around these guys. This one dissociates, that's an H plus, so what's this then? Right. So these are held together, and then I pour that, this is a strong base, sodium hydroxide, so I pour that in the water. What's going to happen with our dissociated molecules? These H's are going to combine with that, right? Because they go back and forth. So what am I doing? Right, I'm taking away the hydrogens, which will increase the pH value. So it's reducing the H pluses. Does that make sense? So, the reason that you need to learn this is because pH is really important to living systems. It's just as important to maintain an appropriate pH as it is to maintain an appropriate temperature. So what happens if you have a really high fever? Let's say you have a fever of 110. So so everyone agrees that you're dead. Well, why do you die? Why do you die? Okay, that's right. So remember, temperature changes the shape of molecules, and shape is really important, right? Well, your proteins, your protein, right? That's what you're made out of. So let's just say, there's tens of thousands of proteins that run your body. But there's one important protein that everybody probably knows about, which is hemoglobin. That binds oxygen. So what happens if your hemoglobin changes its shape and it can't bind oxygen anymore? Hold your breath. It's the same thing, right? So changes in pH in your blood would do the exact same thing. So your blood pH has to stay somewhere between 7.2 and 7.6. If it varies outside of that, it's going to change the shape of your proteins and you're not going to function anymore. And the most important thing in biology is shape. So I can't stress that enough. That is the key. And we talked about that, right? Virus has the right shape of the receptor, it can get in. Insulin has the right shape for the receptor, it can control your blood sugar, and so on and so forth. So shape is really, really critical.
Can I ask you a question? Going back to this H, the reason why it becomes a base is
So yeah, right. So the definition of a base is something that will accept the proton, and a definition of an acid is something that donates it to the solution. So if you're taking it out, you're a base by definition. If you're adding it in, you're an acid. And the only thing that you can do to be a base or an acid is either you give H's or you take them away. That's it.
so yeah, basically binding the hydrogen to itself, so it's not.
Right. So then it's taking it out of the solution. No, because the H's get bound up, right? You can think of it as like, let's say you have X number of hydrogen, you know, the little balls that we messed with in the lab, right? So if someone comes in there and starts taking them and sticking them on their body, then there's no free ones left for you to take, right? In the whole pool of them. So that's what ammonia is doing. It's taking them out, so there's no more left in that pool. Well, it's become positively charged ammonium, and so it's taking the hydrogens out. So this is just giving you an idea of what the various things are. So you know your stomach acid, the pH is close to 1, between 1 and 2. Soda pH is about 3. That doesn't seem like a big difference, right? 2 to 3 or 2 to 4. So that's just 2 points. But really, how many more hydrogen ions is it? So how many more hydrogen ions are in a pH of 4? I'm sorry, pH of 2 versus a pH of 4? 2. Because remember, pH is based on powers of 10. So it's 10 to the second power, which is really, no, 10 times 10 is 100. So there's 100 times more hydrogen ions in your stomach than in tomato juice. What about between your blood and your stomach? How many more hydrogen ions? So what's the difference between pH of 2 and pH of 7? 5, so that's 10 to the 5th. So that's a 1 with 5 zeros after it. So how many more H pluses are inside your stomach than in your blood? So 1, 2, 3, 4, 5. 100,000 times more H pluses in your stomach than in your blood. And so, even though the numbers seem like they're not a big difference, in reality they are, because they're to the powers of 10. Yeah, and that makes sense because when you put carbon dioxide in water, it makes carbonic acid. And so that is an acid by definition. So if I put on the test a certain pH number, you guys will be able to identify if it's base or neutral or acidic. What about pH 8? What's that? Alright, pH 3. pH 7. What's the only true neutral solution? Pure water. Good. Alright, I think you got it. So, yeah, remember pH of a neutral solution is 7. Biological fluids usually are 6 to 8. You know, of course, there's exceptions like your stomach. And that each pH unit is actually a tenfold difference. Alright, so, you know, I get asked this quite a bit from, you know, various sources, but there, like a few years ago, there was this talk about this alkaline diet that is supposed to, you know, help people with cancer. You guys heard of this? So basically what you do is you eat foods that change your blood pH to make it more basic. Right? That's what alkaline means. But what's the problem with that? If you actually were able to change your pH of your blood, like by, let's say, it's a whole pH point, what would happen? It would kill the cancer, but what else would it do? It would kill you, right? So there's no way that could ever happen. Your body has a good system of making sure that you maintain a relatively constant pH. Yeah, so that's a really bad one. Because chemotherapy only kills cells that are actively dividing. Changing your pH would kill everything. like aliens?
No, I mean, like, if someone's really sick because they have acidic blood, like we've had it in the hospital where someone, they couldn't touch them or open them up because their blood was acidic. How would you change that?
So you would have to buffer it, right? So the way that you do that and the way that your body does it is it can convert strong acids into weak acids and their conjugate salts. So a good buffer is like sodium bicarbonate, which is similar to stuff you would take that's in Tums or Rolate. It's actually calcium bicarbonate, but sodium bicarbonate is much more common and more readily available. So let's say that we have um, hydrochloric acid, right, which is found in your stomach, strong acid, and we add this is sodium bicarbonate. We add this to it, that will produce carbonic acid, which is a weak, right? I mean, if I asked you to drink, here's a bottle of hydrochloric acid, and here's a soda. Pick one. Which one would you drink? So you know that carbonic acid is weaker than hydrochloric acid by a factor of 100 or more. So this is changing that pH, right? And then you're producing as a byproduct the conjugate salt, which in this case is chlorine plus sodium, which is salt, right? Does that make sense? So buffers are something that try to maintain a constant pH in your blood and in different systems. And we'll use those in the lab too, because when we work with enzymes, we're going to work with enzymes outside of the cells in our body. So we're going to do like photosynthesis is a good example. So what do plants use for photosynthesis? You guys remember? So what's the equation? Plants take in carbon dioxide and water, right? And then they produce oxygen and sugars, carbohydrates, right? Glucose. So what I told you guys, if you added carbon dioxide into a liquid solution, what does it do? It makes carbonic acid. That's why putting carbon dioxide into soda makes it acidic. So what would happen to the plants then? They're taking in CO2 to do the reaction. What happens to their proteins that do this? Their workers. They would make it more acidic, right? And what does acids do to proteins? Changes their shape, right? And they wouldn't function. So plants, by taking in carbon dioxide, wouldn't be able to do photosynthesis because they would kill all their workers. Do you think that happens? So how do they get around it? It's on the screen. What do? They use a buffer, right? That helps maintain a relatively constant pH. So when you guys go into the lab, we're going to do this experiment. We'll put plants in, we'll expose them to sunlight, but were we just going to put them in straight up water? What do you think we're going to put them in? Water with no. We need to make sure that the pH doesn't change. What kind of buffer? We want a buffer that's against acids, right? What's the one that's against acids? Sodium bicarbonate, which is, anybody know? Baking soda, exactly. So next time you guys go in the lab and we do reactions, the lab techs like to put buffer on there, but if you read the label, it's almost always got baking soda in it, sodium bicarbonate. So we're going to test the enzyme in your mouth that breaks down sugars, carbohydrates into maltose. And what do you think we're going to use then when we do the experiment? to use a buffer, right? Because what would happen if the pH changed the molecule? The experiment wouldn't work. Would it be because the enzyme doesn't work, or would it be because the pH is weird? Yeah. So you have to keep that in mind. Anytime you're working with the protein, you have to make sure that you keep a relatively constant pH. And what else could change the shape of a protein? No. Temperature. That's exactly right. So two most critical things to life: constant temperature, constant pH. Change it, and it's going to change the shape. And if it changes the shape, it probably will kill you. Any questions about that? So on the test, I might ask you, what's a buffer do? Well, I don't even need that specifically. What does a buffer do? What's its job? It resists changes in pH, right? It helps maintain a constant pH. That's it. Are you guys over this? All right. You ready for the next chapter? You know what that one is? This one is organic chemistry. Oh, come on. This is the best chapter in the whole book. Okay, so the word organic chemistry sounds scary, but it's really not. All it means is that it's chemistry of living systems, and living systems are pretty complicated. I mean, we already talked about that, so we need some sort of order. And you guys know from playing with molecules, let's say that those balls and sticks were Tinker Toys, right? You guys played with the oxygens. How many holes are in oxygen? So there's 2 in oxygen. What about hydrogen? And then what about carbon? 4, good. So let's say that I gave you a molecule building set and all I gave you was hydrogens. What could you build with that? So you could just stick 2 hydrogens on a stick, right? That's it. Can you make anything else out of it? No, so then that's all you get to do. Is that a complex structure, you think? So what about carbon? If I gave you a bunch of carbons, you can build all kinds of crazy structures with that. Why? Because there's more bonds, right? So the things that make the most chemical bonds can make the most complex structures. Does that make sense? What can make the most bonds? So let's go over it. How many bonds does this make? 1. What about that? What about that? It's 3. What about that? 4, right? And what about that? 3. And that one? 2. And that one? 1. Next 1. So it's because carbon can make 4 bonds that it's the required molecule for living systems because we can make complex structures out of it. Does that make sense to everyone? So organic chemistry really is just a fancy term for saying the chemistry of carbon, or the chemistry of living systems. Usually that's carbon. It's the ones that we talked about, right? The most common elements. Remember what those are? What are the 4 most, if I ask you on the test, what are the 4 most common elements in, not elephants, elements in, I needed that, in living systems? Oxygen, carbon, hydrogen, nitrogen, that's it. Yep. So that's what we're gonna focus off on in this chapter. All right. So everybody understand why carbon's important? How many bonds can it form? What's its valence? Valence means unpaired electron in its outer shell. What's its valence? All right. So we're gonna start simple. These things, there's no other way to do it. You're just gonna have to memorize it. So we're gonna start with the very simplest carbon. So if I gave you a molecule set and I gave you a carbon, I said build the simplest molecule you can, terminate all the ends on it. What molecule would you put on this? Hydrogen, right? Because hydrogen, once you put a hydrogen on there, you can't add anything else. It can only make one bond. So how many bonds does carbon need? So how many hydrogens would I put on it?
Four.
This is the very simplest carbon molecule that I can make. Carbon plus something else. And that's C1H4. And this is called methane. You guys know what that is? What'd you say? Who said that? It is. That's farts, right? So there's lots of I live in like Chandler, so there's lots of farts going on there from cows. But that's what it is, methane. It's a gas. All right, and then so what's the next most complicated molecule I can do? Least. No, we're sticking with carbons because this is organic chemistry. 2 carbons. So then, what do I have to do here? How many hydrogens do I put on here? Why can't I put on 8? So if I drew this on the test, what would you tell me? Is this possible? No. Why? Too many bonds, right? So I have 1, 2, 3, 4, 5 chemical bonds on carbon. Is that allowed? No. How many can I have? Right. So I got to get rid of that. So you're right. This is C2H6, and this is ethane. All right. What's next? So we got 1, we got 2, now what we need is 3. And so what's that chemical formula? Anyone know this is? I'll give you a hint. Use it to grip. Right, that's perfect. And then the next one. 4. Which one? Oh yeah, so we're gonna get to that in a second. All right, so methane is 1, ethane is 2. Notice they all end in A and E. Right? Propane is 3. Anybody know what that one is? It ends in A and E. I'll give you a hint. They use it in cigarette lighters. Yep. All right, so we could go on, and I could be really mean and make you memorize, 5 and 6 and 7 and 8 and 9 and so on and so forth. But I mean, one of these you probably heard of. It's 8 carbons. What do you think that's called? Octane, right? That's what you read on your pump. It tells you how much octane's in it. That's a 8 carbon chain. So I just want you guys to know the basic four. Okay. Now, carbon, we don't have to limit it to just single bonds. We can put 2 bonds in carbon too. And then if I put 2 bonds in that, is that correct? What do I have to do to fix this? So is that right? Okay, so that has 1, 2, 3, 4, and that one has 1, 2, 3, 4. So that's correct. So that becomes C2H4, and because it has a double bond in it, it's no longer ANE, it's ENE. That makes sense? Right. So ethanol would be ethane with a alcohol, which is an OH on the end. That tells me that there's that functional group on it. But that's getting ahead of ourselves. Let's go back to our ethene. So if I put a double bond there, and obviously I need to erase so that each one of these has 4. What is that called? Good. But what if I put one here? Okay, so let me fix it. 1, 2, 3, 4, 5. Okay. So is that the same molecule? Does it have the same shape? If I had it over here? Right, the shape is the most critical thing. Does this molecule have the same shape as this molecule? Everybody understand that that doesn't? So, what's the chemical formula for this? Okay. Now, if I move this to here, what's the chemical formula? So then how do I distinguish one from the other? Yeah. So he's right. So we use numbers. So this would be 1 butene because we start numbering the carbons like another color. So we start numbering the carbons. Like so. Did we get that? So because that double bond first appears on the 1 carbon, we call that 1 butene. Now, if I move it here, and I know I'm not changing the hydrogens, but you guys know where they go. So what would you think that would be called? And if I moved it over here, yeah, or you could put it in front of usually you put it in front of the name. So there's a standard nomenclature. It's called IUPAC, and I don't care that you know that, right? But there's a particular naming system that you have to follow.
So,
But this isn't 3. Go ahead.
let's say someone just does the formula C4H8.
Mm-hmm.
We don't know where it's double bonded.
Right.
Would we just say it's butene no matter what?
Well, you could, but that wouldn't be exactly accurate, right?
Right? 'Cause
I mean, it's not specific. That's like saying, okay, that's a car, not, that's a Tesla S or something like that.
Would it be an unknown mutant? Would that be accurate?
Well, I mean, if you don't know, then you don't know, right? But, it's just like,
So you're saying there's a specific notation that
right, there's a specific notation you don't need to worry about. What I'm trying to get at here is the reason that this is important is because later on, these numbers are going to be important when we talk about this molecule right here. You guys know what that is? This is a super important molecule. We decide that all living things have to have this.
It looks like oxygen.
It is. This is ribose, which is RNA, and this is, so ribose is OH is there, and so we have to number the carbons because we have to know where everything is. So we number it clockwise, 1, 2, 3, 4, there's another one here, so we number it 5. So we know where things are, right? So the base, the letter A's and G's and C's and RNA, there's only U's. But if I wanted to turn this into DNA, I would make it, what does D mean? What does that mean? No, that's die. What's D? What if I depant you? What am I doing? So that is deoxy. That's DNA. Deoxy. I took an oxygen away, right? What did I take it away from? The number 2 or the number 3 carbon? So, you have to be specific. So this is why it's important you guys understand this numbering system. I really don't care if you know it for any other reason except for DNA. But this is critical because you need to know where the base is, where the phosphate backbone is, it's on number 5, and so on and so forth. Because this matters when we talk about how DNA is copied. And you know that that's important because you have to copy your DNA to make new cells, you have to copy your DNA to make new offspring, to make gametes, sperm and egg. So that's why this is important. Right? I could care less if you know how to number, 183 octane or whatever. But I want to go back to this. So you guys told me that that's 3, right? But remember, these are molecules. They're not stuck on this board. I could rotate that. So if I rotated this, where would that bond be then? So then there's no such thing as 3, because that is really 1. And so that's why it's important you guys play with these models, because you have to get it in your head that you can rotate these things. They're not stuck on a flat surface. Everybody understand that? Okay. So methane, ethane, propane, butane, you're just going to have to memorize them. And then if it says E and E in it instead of A and E, what does that tell you? It's got a double bond, right? And we have to be able to tell the location of the double bonds because where that's located makes that molecule have a different shape.
So if it's on 4, would it be 2 then?
It's not, it would never be on 4, right? Because that would rotate and it would be on 1. We always number from the lowest numbered carbon. So even though it's between 2 and 3, that would still be a 2. If it's between 1 and 2, that's a 1. Make sense? Okay. Everybody know the valence of all these molecules, I'm sure. Hydrogen can make how many bonds? Oxygen, nitrogen, carbon. So I've already shown you that we can vary the carbons in length, right? 1, 2, 3, 4, and so on and so forth. But we can also vary their shape. So let's I'm going to get rid of all of this. I'm going to focus on this molecule here. I don't Everybody got it? So molecular formula of this is C4H10. Everyone agree? Okay. So what about this? What's this then?
See,
How many hydrogens? Same shape? Then they're different. This one is actually called, and it has different chemical and physical properties, just like we talked about sodium, right? Explosive metal, chlorine, deadly gas, put them together, different shape, different chemical, physical properties. This is different than that. But could I just write this out? Just be like, this, here's some C4H10, go ahead and take it. So you have to specify. So this, the term for this is an isomer. Iso means same and meros means part. So these are made of the same parts. Alright, so I can make straight chains, I can branch them, you know, heck, if I wanted to, I could link all these together in a circle and make a ring out of it. So there's all kinds of different shapes that you can do.
but they would all be different.
Right, because it's got a different shape, they're different molecules, and that's important. And you know we can change the number and location of the double bonds, and so on and so forth. So we can vary the carbon skeleton, and I think that you guys get it. Ethane, propane, different links. Here's butane and isobutane. We talked about 1 and 2 butene. Why isn't there a 3? Because that can rotate, right? And then these are what we call cyclomolecules because they have carbons and ring structures. All right. So this kind of isomer, which means same parts, right? This is a structural one because the structure is different. Did we get it? But there's another kind called the geometric. So here's a structural one. These are different. Their chemical properties are different. Their physical properties are different. So the next one is called the geometric, and it's because the geometry of it is different that you can have different kinds of molecules. So let me ask you this. Is that properly drawn? Can that molecule exist? Does everything have the right number of bonds?
We each the other carbon to have.
Well, okay, just this side. So let me fix it. Then I'll re-ask. Is that drawn correctly? Okay. So I've drawn this molecule. I'm gonna draw another one. 'Cause I like drawing molecules. Are those the same? So think about when you guys were building the models, right? If you had one bond, could you spin the ends? So if I took this and I spun it right there, would it look like that? So then, are they the same? The answer is yes. Now,
So are they continually spinning that, or is that change?
well, yeah, they can rotate just like, you know, if I stuck 2 oranges on a stick, I could spin those all day and night.
And it changes that shape, it's gonna change its, as you said, the shape.
Well, you know, it can rotate and combine with other things. So it really depends on what kind of rigid shape it has, what kind of fixed shape it has in the molecule. Alright, so you got to keep that in mind. Now, I'm going to change this by doing this. Now, are those the same?
No.
Why not?
Because you can rotate.
Right, so if I have oranges with two sticks in them, or you guys built these models, right? You had the oxygens with the double bonds. Could you spin those? No, because the bonds would hit each other, right? It would prevent you from doing that. So in this case, these are different molecules, right? These are geometric isomers. What's the formula for this? Right. And what's the formula for this? So they're the same, right? Same parts. Different arrangement, different shape, then they're different. Shape is the most important thing in biology. Alright. So there's terms for this. If you're going on a voyage across the Atlantic Ocean, what kind of voyage would that be? So the word for across is trans. This is a trans molecule. This, you probably don't know this word, it's not used very often. But this is called cis because it's on the same side. This one is called trans because it's on opposite sides. So you guys probably heard in the news, what's the kind of fats you're not supposed to have? What does that mean? Yeah, so what's a trans fat? What does that mean? Why is it bad for you? Okay, so here's this is a fat, right? It's a fatty acid is just long chains of carbons, like so. So we can have long chains of carbons, and in the middle we have a double bond. So that molecule can continue on like this, and then up here we would have hydrogens, right? Or it could do this. So what kind of fat is that? That's a cis fat. What kind of fat is that? Trans fat. So if we just ignored this, what shape does this have? What shape does this one have? So trans fats have different shapes, but why are they so scary? Why are they bad for you? You guys ever had a snowball fight? So do you just pick up the snow and throw it at people? Why? Well, what does that do? Yeah, what's the word that we use if you put more material in a smaller space? What is that? Well, we call it dense, right? So what do you think that I could, if I was stacking fats and I was stacking them in a jar like this, what do you think I could fit more in? Ones that are flat or ones that are U-shaped? So what happens when you compact something? What does it do? It gets more solid, right? So these fats, because they can be more compact, they're more solid at room temperature. These are more liquid at room temperature. What would you rather have in your arteries? Something that's solid at room temperature or something that's liquid at room temperature? So that's why these are not so good, because they tend to be more solid at room temperature. Does that make sense? So that's where the word trans fat came from. Didn't know that, did you? You learned something new. All right. So the last one's a little weird, and you're gonna have to kind of take my word for it. But you guys probably forgot this because we did this lab so early. I really wanted to do it this week, but carbon can make four bonds, right? So I don't know if you guys remember this or not, but you built amino acids. Do you remember what the groups were on there? So one was an amino, which is an N, H, and H. Remember I asked you guys to identify the functional groups, but you forgot. And it had a hydrogen on it, and they had another functional group called carboxylic acid, which is C double bond O O H, like so. And so this word is amino, and this word is carboxylic acid, so that is called an amino acid. And then this varies, right? It could be a ring, it could be just a hydrogen, it could be an OH, it could be a methane. Right? That would be linked there, so we'll just have three hydrogens on it. We call that a methyl group. But there's 20 different things that can go there. And I don't care that you memorize them or not. But what happens is in chemistry is that if I have 4 different groups on a central carbon atom, it makes something handed. So it makes something that's like a left hand and a right hand. We call that chiral. For handedness, that's the word. If it doesn't have that, so if this were a hydrogen, would that have 4 different groups on it? One, those are the same, so that's 2, that's 3. So this wouldn't have a left and a right-handed form, and we would say that's A, which is the word for not, chiral, which is handed. So this would be not handed. Right? So in this thing, it's handed, and so we call those enantiomers, or the other term is optical isomers. Which one? This one? Because we have hydrogen, hydroxyl, carboxyl, and amino. If that was a hydrogen, then I would have amino, hydrogen, hydrogen, carboxyl. So that's 1, 2, 3 groups. Does that make sense?
So, what I'm confused about is when you put the oxygen,
Right.
both hydrogens are no longer in some groups. With the oxygen, then the hydrogen by itself will block.
Well, it doesn't matter. Let's say I do this and I do that. There's still only 3 different ones. Or I could do this.
Okay, so they have to be different.
Distinctly different. All right, I'm going to show you why. So it's really, this is really difficult to visualize. So you kind of got to play with models to see that this is really a true effect. And it has to do with, moving them around in a 3D space. So it has to do with the complexity of the molecule too. So let's look at, hopefully this will play. Well, let's look at something simple like a pencil. Maybe it's not going to play. Oh, now it's gone. Okay, so I can do this demo up here. So if I put a mirror in between these, they would be a mirror image of each other. Everyone agree with that? So I took the mirror away. I could make this be the same as that in the same plane if I slid it. Does that make sense? So in this case, there's not a left or a right-handed form because they look the same, right? My hands look different on the same plane. Oh yeah, I could do that, but that doesn't count, right? If I wanted this hand to look like this hand, I'd have to cut off this thumb and glue it on the other side. So that's what we mean by handed. So pencils, they're not handed because they're not very complex. But things like hands, they are. And everybody gets that? Okay, now let's go to molecules. So achiral means it's not chiral. Remember I said if there's three different things attached to a central carbon, then it's not handed. So here's an example. We have, this is, I believe this is nitrogen. I'm not sure what the green is, but I think it's nitrogen. This is oxygen, right? That's hydrogen. So how many different groups on here? No. 1, 2, and those are the same, so that's 3. So this isn't handed. That's a mirror image, right? There's a mirror in between there. They look exactly the same as they would in a mirror. We agree with that? All right. So when I rotate this, that's not me, but let's just say when it is rotated, are those the same? Yeah. Oh, everything lines up, right? So that means that it's not handed. It doesn't have a left or right-handed form. Well, they're both the same. So they're not isomers. Remember, isomers are the same parts, different shape, different chemical property. Now, if I have 4 different things on there, that makes it chiral, which is handed. So mirror image, right? Take the mirror away. Still mirror image. But when you rotate this molecule in the same plane, it's not the same. Do you guys see why? This one's red and that one's white. For them to be the same, they would have to be the same color at the same location. And there's no way you could rotate this molecule and make them look the same. It's not possible. Well, okay, but you can't do that. It's a rule, it's a bond. You can't just, it would be like me cutting my thumb off and gluing it on the other side. Right? So this is just, it's part of life, and when drugs are produced, sometimes they have a left and a right-handed form because of this. Right? So you guys know Prilosec and Nexium? So those have a left and a right-handed form. Prilosec is a mixture of both, and Nexium is just the biologically active form. You guys ever go and buy amino acids? So remember I told you amino acids has a hydrogen here? And then they can have variable groups over here. So do you think amino acids have a left and right-handed form? They all do except one. The R group is in hydrogen. But, let's look. So a common amino acid would be like tyrosine or something like that.
Well, they're different, so that one that you have up there is, that one's a chiral.
Right, so it would have a chiral form. So how do we distinguish one form from the other?
Because they don't line up.
I know, but I mean, when we were distinguishing butane from the other form, we called one isobutane, right? So how do we distinguish one-handed form from the other? And we just give them different designations. So in biology and science, we call the left-handed form the L form, and the right-handed form So it has to do with the rotation of the bond, and I don't really care that you know that. I don't want to get too deep into this, right? I just want you guys to know that there's a left and a right-handed form, right? And that's important. So if we look at like the amino acid tyrosine, what are they selling at Amazon? Which form? What does it say in front of it? L, right? In fact, if we went shopping, it won't let me. It's blinking. You have to wait a second. Everyone, look over here. Yeah, I was told that we're getting laser projectors, and so they are going to replace these or whatever, but they don't want to mess with them. So I don't know. I'm not the media services people. I just report the issue. But can you guys over here see what that says? So what's the letter? You guys help them out. What's the letter in front of it? L, right? So which form is the one that they sell? So why don't they sell this form? Okay, so but why don't they sell that? That's exactly right. In fact, it doesn't work at all. So if someone came up to you and said they wanted to sell you D-tyrosine, what would you tell them? Go take a hike. It's not going to do me any good. So your body can tell the difference between a left and a right-handed form of a molecule. That's pretty freaking amazing to me. And when you're giving drugs like Prilosec and Nexium and things like that, those almost all the time when you're synthesizing them, they have a left and a right-handed form. So let's look at what's the word for what's the scientific, the medical chemical term for Prilosec? Stop flashing so I can turn it back on. It's omeprazole. So in medicine, Prilosec is a mixture of, remember it's medicine, so we use S and R. So it's a mixture of both of these. Do you think they're both work to block the proton pump? No. Which one do you think works?
So why do they do the mix? Just because during the process it makes both of them?
Yeah, so if you're an organic chemist and you're synthesizing these things, they come out in equal parts. There's nothing you can do about it. The only way to make them purified is later on you purify out the. You're right, it's cheaper, and also there's a little trick. So Prilosec was the first one, right? And the chemical companies know this, the pharmaceutical companies know this. So when they first synthesized it, they got FDA approval to sell it, and they could sell it because they had a patent on it for X number of years. And then where did it go? It goes over the counter. Then they synthesize the other kind. It's a different chemical altogether, right? So they get another patent on it, and now all of a sudden they can sell those at behind the counter prices instead of over the counter prices. Does that make sense? Right, so there's different doses because you get twice as much of the medicine for the same amount of mass. So what is this called? So remember Prilosec was omeprazole? So this is, the ES is a little trick, a play on words, because this is what it is. S, the left-handed form, which is the biologically active form. This one doesn't do anything. In fact, sometimes when they synthesize these together and they give it to a patient, sometimes the biologically active form does what it's supposed to, and the other one can have bad side effects, like Prozac, for example. So the half of it was a dopamine reabsorption blocker, and the other one caused sexual side effects. So do you think the pharmaceutical company cared about that? So So after their patent for Prozac expired, what do you think they did? They purified it into the, and then they kept selling it as a prescription medication.
So for one version of medicine that uses scientific maybe it's a little before they use L, I'm sure they're using
Yeah, so that's like that's a science term. Like if I bought chemicals, like there's a form of, and we'll talk about this later, but there's a form of glucose called, it's the D form. So it's D glucose, or it's also known as dextrose. Right? Your body doesn't use that; it can only use the L form. So they use it as a filler in like Splenda and stuff because that stuff's really sweet. So there's certain nomenclature that you'll see for like chemicals that you buy versus drugs that people are going to take, right? Because the amino acids aren't really regulated.
So esomeprazole was sold second, right? So that's
Yes.
more purified stage.
Mhm.
So that's what is really gonna work for you.
Right. That's the only form. Even in Prilosec, it comes in a mixture of S
But you
and
only
R
use
forms.
the S.
Your body will only use that one.
And that
The other one
cause
either
that.
may not do anything, or 9 times out of 10, it'll have some sort of negative function.
So do they do that just to make sure that they continue to have a patent later? Do they
Well, I'm not in the war
Why-
rooms whenever they make these decisions, right?
why do that? Why not just come out with it more purified?
Because it costs more money, and they can sell it to you anyway. I guess. I don't know. You have to ask. Call up, Mark or something, and ask him. Hey, what's going on over there?